Well, good morning. Welcome to our webinar on using big data to address challenges in autism research. My name is Joanne Geer. I'm the Executive Director of the Biopharma Research Council. We're an association for scientists across all the many silos of biomedical research, which includes industry, academia, nonprofit, government, and suppliers, and those who love them. We have a number of activities coming up this year, and I'm not going to go through any of them in detail, but if any of these intrigue you and you have any questions, we really appreciate you being in touch with us. Our organization is about 7 years old now, and we reach about 9,500 scientists on a regular basis. I'm just going to say a little bit about this program. It's the first time we're doing a big expo, which will be at the Doubletree Hotel in Somerset on November 17th. And it will look at every aspect of pharmaceutical manufacturing. And we're very much seeking potential partners, people that would like to contribute to the program, and so on. Throughout this time, please enter any questions you have in the question box on your dashboard. We're going to hear from our speakers first, and then we're going to open up for Q&A. So if there's a question about a particular slide, it's a little bit helpful if you mention that in your note. And here we are. I'm going to introduce both of our speakers, and then we'll hear from them. First, Dr. Alicia Halliday. She is the Chief Science Officer of the Autism Science Foundation. She holds a PhD in psychology from Rutgers and has worked previously with the National Alliance for Autism and Autism Speaks. Our second speaker is Dr. Elise Robinson. She is an instructor in medicine at the Massachusetts General Hospital. Her work has linked genetic risk factors for severe neuropsychiatric disorders to variation in behavior in the general population. Dr. Halliday.
Okay, great. Oops, I'm sorry, everybody. I thought this was already open. Here it is. Sorry. Okay, here we go. Okay, so hi, everybody. I wanted you to be able to put a face with a name. The picture on the top is me in the middle and my two daughters. They're twins. They're 5½, and actually one of them, the one on the left, was diagnosed with autism spectrum disorder a couple of years ago. So she's doing great, and we'll actually talk about the different types of autism that's considered to be on the spectrum and why big data is actually needed to solve the problem. So in case you don't know exactly what autism is, it's actually a group of features that has been kind of reclassified and is rethought every few years. So some of the core deficits are in social communication, and that includes deficits in a social emotional reciprocity, or the ability to kind of communicate with others and understand things like facial expressions. There's also deficits in nonverbal communicative behaviors, so things like gesturing or expressing intent have deficits but aren't always gone. There's also deficits in developing and maintaining relationships. And then the second core feature is restricted and repetitive behaviors, and these can be anything from stereotyped repetitive motor movements, hand flapping, or stimming. It can also mean insistence on sameness, which means people with autism generally don't like a whole lot of change and like to have things in a particular order and for them to stay in that order. There's also restricted fixated interests. The most common of these you see in boys, where they're kind of fixated on, instead of the toy itself, the wheels of a toy or the buttons on a computer, and these also are different from boys to girls. And also hyper or hypoactivity to sensory input. Hypoactivity is somewhat rarer, but hyperreactivity is more common, where an individual is very sensitive to lights or sounds. So these are two people on the right who are both diagnosed with autism. I don't know if you've ever heard of her, but Temple Grandin, who's on the top right, is actually a PhD. She's someone who has experienced challenges with autism, but certainly has very, very high cognitive ability. She has a very high education. She travels and she works and is very productive at animal husbandry. On the bottom right is Jody. Jody lives at a residential care facility. Jody has minimal language and has deficits in intellectual functioning. She has some severe repetitive behaviors that have included things like head banging and self-injury. So why is autism a public health problem? As many of you have heard, the prevalence of autism is 1 in 68, or 1.5% of children measured in this study in 8-year-olds by the CDC. Not only are they those that have above average intellectual ability, of those 1 in 68, about a third of them have an intellectual about a third of them have an intellectual disability. Another group have normal intellectual ability, and the other ones are in the borderline range. So you have a really big spread here of people with different levels of ability and disability. And so part of the purpose of this webinar is kind of to try and figure out why it's taking so long to figure out what causes autism, or what specifically causes autism, how to treat the different kinds of autism, and how to actually understand those different types of autism and understand the strengths and uniquenesses and weaknesses of people across the spectrum. So to give you an idea, this is a slide that was taken, I'm not going to claim credit for it, it was taken from a group in Australia, but I did enhance it with some photos. So as you can see, if you look at and I'll just move my mouse, the domain of communication, you have people who are nonverbal or speak or have minimal verbal abilities, to people who are very verbal, who can speak. Maybe they have a different style of communication, but they certainly can speak. In terms of social interaction, you have those who are considered aloof. So as you can see in this picture, this boy in the tree, these two boys are not really playing with each other. One of them is kind of ignoring the other and kind of in his own world. On the other hand, you have, and I know this is kind of stereotypical, you have the active but odd, and this is a character from the Big Bang Theory who many people have hypothesized is on the spectrum, but he is obviously someone who is very, very, very gifted, and also but has an odd communication style. So in terms of repetitive and restrictive behaviors and sensory processing, you can have those all the way from hand flapping, which is very mild, to head banging and skin picking, which can be very severe. On the right, you see Jackson. So Jackson has, I would say, in the middle between mild and marked. He, every time he sees a candle, he has to lick the candle, so that's his repetitive behavior and his restricted interest, one of many. And he also is very hypersensitive to noises, and so a lot of kids with autism actually wear these sound mitigating headphones. So that's an example of two, extreme behaviors. So another one is the learning style, and this is actually for those that that actually show improvements in learning over time. And then, so some of them have strong visual spatial cues and are very visual learners, and some of them are more detail focused and can't see the big picture because they're looking so tightly at the details. And then in terms of intellectual disability, we talked about this before. As you can see, this boy on the left, he actually is severely affected and has a very low IQ. And then you have individuals all the way to who are considered above average or gifted. And on the right is Kerry Magro. He's an activist. He's an author. He's a consultant. He started his own philanthropy. So here's someone that has excelled in the community, even with autism. So how do you put all of these different types of autism together, or how do you differentiate them out? One of the ways we know that autism is highly genetic. Individuals who have an older sibling with autism have a 20-fold greater chance of having autism themselves. Parents of individuals with autism commonly have not autism itself, but autism-like symptoms. Ways that researchers have really tried to parcel out what causes autism and to kind of enhance better treatments of autism is through genetics. So researchers now know that for some of these rare things, that there are genes that definitely contribute, that definitely cause autism. These are genes that are associated with disorders like Fragile X, Phelan-McDermid syndrome, even epilepsy, that if a person has this, this gene or this gene mutation, the probability is very high that they also have autism. There's also about half of all the autism genes are probably common, so everybody has them, but in their right combination in different mutations, that's when they lead to autism. And 41% of autism is unaccounted for, and it doesn't mean that it's not genetic, it just means that we don't know yet. Excuse me. So actually, this particular slide shows that exactly what I said. If we don't know all the genes that have caused autism. But if we get better and better at finding them, you can get up to 1,000 genes that are associated. You could just say these are genes are associated with autism with the more cases you have. So the more people we look at, the more genes we're finding that are associated with autism. And again, these aren't genes that are causative per se, but they're definitely contributory. So as I just mentioned, it's a mixed bag. So if you're a person with a low burden of common risk variants, so you have, say, if there's 200 of them, and that's just a sample number, if you have 10 of them, then you probably have a low risk. But if you have, a high number of them, then you have a high risk of having autism. So there's everything in between. And the point of this is to kind of explain that we need lots of people to study and lots of data from those people to understand what the different influences are. And I can't just talk about the genetics without kind of mentioning that the environment is also kind of an open question right now. And I don't want to give anyone the idea that the environment is just toxins or chemicals. We think of the environment as everything from parental age to medication during pregnancy to even things like stress. And also one of the big environmental factors is shown to be immune challenges during pregnancy. So having a parent, if you're a woman and you get a severe illness like the flu or even a severe infection, either bacterial or viral, your chances of having a child with autism are higher. And then also there are also things like air pollution. And honestly, researchers no longer believe that that unknown group is just genetics or just environmental. Most scientists believe that it's a combination of the two, that there's a group of people that it's not just genetics and it's not just environment, that it's a combination of the two. And again, to figure out what the different combinations are and what are the gene susceptibilities and what are those environmental factors, you need lots of people to study. So these are the sorts of things that there are differences across. We already mentioned verbal ability, cognitive function, also the age at first symptom presentation. Typically you can see the signs and symptoms of autism by 18 months to 2 years, and a lot of people can make a diagnosis by 2 years, but in some cases they may be kind of borderline and then they don't reach criteria until about 3 or 4 years. Again, we mentioned the known genetic causes. There's also quite a number of comorbid medical features. Kids with autism have, or individuals with autism have, a high rate of gastrointestinal problems, a high rate of epilepsy or seizures, and then a very high rate of sleep disorders. But you don't have to have all three of these. So there are individuals with just GI problems or just sleep. And also there's big differences across gender. So there's a lot of things, and we could go into a whole other presentation about gender, but because there are more males diagnosed with autism than females, there are more males to study, and one of the challenges of research is now to understand females with autism better. So some of the questions about autism are: what causes it? How is it diagnosed? How do you treat it? And what do people with autism need in terms of services? And again, these are all questions that can be answered with big data. I also wanted to point out that big data isn't just important, and I'm sure everyone on this webinar knows that. It's not just important for understanding the causes, but also how to treat autism. So in this left panel, you can see the different features of people with colon cancer. And if you gave them all the same therapy, you would have some of them that did well, some of them that didn't have an effect, and then some of them that actually got worse over time. You can take the same, the same kind of analogy to autism. If you give everybody with autism, with all the different types and all the different features and all the different behavioral symptoms and all the different genetics, the same therapy in the same way, you're going to have some that do well, some that don't, and then some that actually decline. The same thing with autism. If you were to able to separate them either through, DNA, through some sort of behavioral subtyping, you could come up with more targeted or personalized interventions for each group, increasing the probability that everybody will benefit from that intervention. So just to give you an idea, there are ongoing efforts in autism research to understand big data. One of them is the Autism Sequencing Consortium, and this is a group of smaller genetic studies that are all coming together to contribute data and behavior information into one giant repository so that individuals, researchers, can go in and tap into that resource and get access to large groups of data. And frankly, this is kind of the way that most of the major genetic findings over the past few years have happened. So these are some examples in the middle. They include genetic projects, and actually this is not even all of what's in the Autism Sequencing Consortium. I just thought I'd give this as an example. So some that just have genes and behaviors, some that has environment, some that look at very, very early signs and symptoms, all the way to 6 months of age without a diagnosis. So here are some basic questions we still don't know about autism, all of which can be addressed through big data. I already mentioned why are males more commonly diagnosed than females? What genes that have been identified in combinations of genes correspond to what symptoms of autism? Also, is autism part of a bigger spectrum of mental health issues? So is autism part of this broad continuum of things like OCD, ADHD, and schizophrenia? Certainly we know there's high tie-ins between autism and intellectual disability and autism and epilepsy, but they could all be kind of one big, gamish, and autism is just one part of that rather than being so specific to being autism or no autism. And then along with what genes correspond to what symptoms, what treatments work best for what symptoms and in what combinations, including can autism be managed through personalized medicine. So actually, the other speaker, Dr. Elise Robinson, is the best person to speak on these ideas. So I'm going to go ahead and stop and allow her to take over.
All right, great. Thank you, Alicia. I'm very happy to follow that since it was such a direct and fantastic introduction to the idea of heterogeneity and what I'll be talking about. So I am a genetic epidemiologist and psychiatric epidemiologist by training. I'm mostly interested in using tools that are derived from very large genetic studies of autism to better understand the manner in which genetic risk factors are creating risk for disease and the type of phenotypic risk being created. So in other words, using results from these very, very large genetic studies of ASD to better understand autism itself. There you go. And I'm going to start by going through some basics of the genetic background of autism-related neuropsychiatric disorders and get us to the types of tools we now have available to us, the types of genetic tools now available to us because of big data. So to begin, neuropsychiatric disorders, including autism, are highly heritable. And that means that a large contribution of risk to becoming a case comes from genetic variation. And on the upper right part of the panel, you see, or the upper right part of the slide, you see a little figure with lots of green dots. And ASD and schizophrenia are hovering around this 0.8 line of heritability, which is suggestive that vaguely 80% of case control variation in these phenotypes comes from genetic differences between cases and controls. So neuropsychiatric disorders are as genetically influenced as a lot of other conditions that that in earlier days people initially focused on, like breast cancer and type 2 diabetes, in which the genetic factors seemed a little more obvious. So neuropsychiatric disorders, particularly schizophrenia and autism, are also associated with substantial loss of fecundity, and that means that people who have these disorders are less likely to have children than people who do not. Now this is a a figure from a study a few years ago in JAMA Psychiatry using Swedish registry data, which compared the number of children on average had by a person with neuropsychiatric disorders to their siblings who did not have that disorder. And if you look at autism in both boys and girls, which are the third and fourth ticks on that x-axis, boys with autism, on average, per this study, had about 75% fewer children than their unaffected male siblings, and girls with autism had about 50% fewer children than their unaffected female siblings. This is a very strong impact and suggests that the genetic influences on autism are strongly selected against in the population, which effectively just translates to saying that evolution does not like them and they should be depressed in the population. And this results in an inevitable association between the degree, the amount of risk that can be created by a genetic variant in terms of its autism impact, and the commonality of that variant in the population. It basically means that large effect alleles that create a large amount of risk for autism, have to be rare, because an allele that creates a large amount of risk for a disorder associated with significant loss of fecundity is pushed down quickly by evolution over time. And that means that we effectively have two tools that come out of genetic studies. The first are GWAS studies can associate common variants of, or genome-wide association studies, that is, excuse me, which are studies of common variation across the genome and its association with disease. Genome-wide association can yield associations between common genetic variants of very small effect size, because in order for those alleles to create risk for autism and remain common, the risk they confer to disease has to be tiny. And because the risk conferred by common alleles has to be tiny, it's taken a long time to start to find them for autism, because you need very big data to associate genetic variants of small effect with disease. So recently, nearly 2 years ago now, a remarkable study of schizophrenia associated 108 common genetic risk variants with schizophrenia risk. Until very recently, in data that have not yet been published, ASD sample sizes were less than 1/10 available for those for schizophrenia, and the number of discovered loci was about the same. And so the difference in discovery for a disorder like schizophrenia and a disorder like autism, at least at present, is not necessarily related to a difference in genetic architecture or a difference in the average contribution of common genetic variants. It's simply a difference in the available sample size. And as the consortia working on autism GWAS grow, you'll likely, almost inevitably, see a very similar uptick in the number of associated discovered loci. So big data are absolutely necessary for discovering common variants associated with autism. What you can do with currently available sample sizes is build so-called polygenic risk scores for lots of different disorders, where any given common variant must, because of its frequency, confer a tiny little bit of risk for ASDs, but those tiny little risks add up across the genome and create what we refer to as a polygenic burden. And that's not unlike height or IQ or other things that we understand to have risk factors distributed across the genome that work together additively to create, for example, how tall you are. There's no one risk allele for height. There are many, 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 many thousands, and they operate additively. You can do the same thing with schizophrenia risk or autism risk. You take the additive contribution of these tiny little effects, and for any given individual can create a polygenic risk score that puts a number on their genome-wide common variant risk for autism. I'll be discussing how we use those a little bit later. The other option is rare genetic variants, very rare genetic variants, can create very large effect sizes or can be associated with very large risk for autism. But in order for us to find variants of a very large effect, effectively have to be new to the population. And that's why there's been a very long-standing interest in de novo mutations. De novo mutations are genetic variants that are seen in a person but not in their parents. That means it's a new mutation to the family and often to the population. Because selection has not had opportunity to act on those variants, they can create very large risk for autism. And through trio sequencing, which is the exome sequencing, historically has been the exome sequencing of a parent, two parents and a child, over 70 genes have been associated with ASD risk by finding these large impact de novo variants. So in constructing genetic tools from big data discovery, we have two primary options. We can look at polygenic risk, which is the additive contribution of tiny little effects across the genome that's inherited, and it's found in all of us at some degree. We all lie somewhere on a continuum in terms of polygenic risk for ASDs or schizophrenia or lots of different disorders. Or a de novo variant risk. De novo variants are not a quantitative score. It's simply a binary variable. Do you have one or do you not have one? And importantly, all of us have de novo variants. Few of us have de novo variants that are strongly associated with autism risk. Okay. And we're interested in using these tools to begin effectively deconvoluting the association between genetic risk for neuropsychiatric disease and those disorders in the population. Is in the traditional manner that we study them, we lose a lot of insight into phenotype by simply saying autism or no, or schizophrenia or no. And a lot of that comes from the degree of heterogeneity that Alicia was referring to. There are likely many different, at least partially etiologically distinct disorders lumped into the autism bucket, and we can use genetic tools to start pulling them apart, identifying populations with the ASD that are more etiologically similar, which is important in the long run because those populations will also probably be better isolated testing populations for therapeutics. The more similar we can make our populations in terms of the etiology underlying the disorder, the better luck we should have testing an appropriate therapeutic. I'm going to talk a bit about heterogeneity in that sense, and I'm also going to talk about the relationship between genetic risk for ASDs and variation in the population in terms of traits of autism spectrum disorder. And that's another concept that doesn't fit well within the DSM model. Typically we think of these disorders as binary things one either has or does not have, but that eliminates a lot of variation in the population that exists in all of us. People do vary substantially. Controls, people who do not have a diagnosis of autism, vary enormously in their social communication ability, and we've wondered a lot about whether any of that variation relates to genetic risk for autism. So I'll run through this quickly because Alicia covered it so well. There's been a huge amount of change over the last 20 years in particular in terms of the average face of autism, and one of the clearest changes that has come with the diagnostic expansion that relates to the prevalence increase is the decreasing frequency of intellectual disability in cases. 30 years ago, between 75 and 80% of people with a diagnosis of autism also met criteria for intellectual disability, which is to say an IQ below 70 or an IQ that's more than 2 standard deviations below the mean. By recent estimates, that fraction is in the low 30s. That is a massive average shift in the average cognitive profile seen within autism. And this matters because we have reason to believe that cognition and functioning, defined various ways, and neurodevelopmental complications are likely to relate to the type of genetic influences that are most often creating risk for different sorts of cases. Now, going back to this figure that Alicia showed, we want to know how consistent is this breakdown of genetic influences between different types of autism cases. Are cases with very low IQ likely to have the same chance of seeing a contributing de novo event as cases with high IQ? Are cases with seizures going to have the same breakdown of common and rare variation contributing to their risk as cases without seizures? Things like that. So beginning with de novo mutations, de novo mutations have been strongly associated with ASDs, as I mentioned, and one of the class that we're most interested in is this PTV class, which I just circled with my mouse, protein truncating variant, which is another term for loss of function variant, which is used equally commonly. And those are significantly overrepresented in people with autism, so it makes us think that as a class, having a de novo mutation in the coding region of your genome that results in the loss of function of a gene is associated with autism risk. What those numbers mean are that about 14.5% of people with autism have a de novo mutation that results in the loss of function of a gene that is protein truncating. About 9% of people who do not have autism have a loss of function mutation somewhere in a protein coding gene. So two things should be taken from that. One is that the class on average is associated with risk. And the second is that we can't say that any given mutation, with the exception of those that are found in significantly associated genes most often, is creating risk because there is this background rate. There's a neutral background rate of mutation that exists in the population. And so not all de novo loss of function mutations will be creating risk, even though they fall within a category on average that does. Okay. And one of the consistent findings in the ASD genotype to phenotype world is that cases with low IQ have higher average rates of de novo mutations. This blue line here, the diagonal one, is the association between de novo loss of function rate in cases and IQ in cases. And what you see is that cases with very low IQ have de novo loss of function rates that are similar to those seen in severe intellectual disability. And ASD cases with very high IQ have de novo loss of function rates that are similar to that seen in controls, which suggests that on average there is a substantial difference in etiologic architecture across different types of ASD cases. And it also suggests quite strongly that on average IQ loss is associated with the de novo influences on autism risk. This is a picture of the distribution of IQ in ASD in one of the data sets that Alicia was mentioning. And it's just showing that in addition to ASD cases having IQs that are below average, they're centered around between 85 and 90. Here it's about 90. There's also expanded variance of this distribution. So it's not just shifted down, it's wider. So there's more high IQ than you would expect by chance, about 6 times as much of it, and more low IQ than you would expect by chance based on that mean. And in terms of classical genetic theory, that suggests to us that there's heterogeneity in the genetic influences in ASD, specifically as it relates to cognition, in that some of those genetic influences on ASD risk are going to be on average pulling IQ down, and others are on average going to be IQ neutral or pulling it up. And it's just another metric of heterogeneity and another reason we need big samples to figure out what's going on. And there's emerging data that suggests that this is actually going on. So in now more than 7 studies, the inherited genetic influences, the common polygenic influences on autism risk have actually been associated with higher IQ and more educational attainment in the population. And this is developing, but it's certainly a recurrent and highly repeated finding at this point that will require a lot more investigation. So secondly, I'm going to talk about thresholding and how we can use genetic tools to understand the relationship between autism and categorically diagnosed neuropsychiatric disorders more broadly, and more typical differences in behavior and cognition that exist in everyone in the population. So there's a very old idea that people, particularly family members of affected individuals with ASDs, often display sub-threshold traits of autism spectrum disorders, meaning people might not meet full criteria for an ASD diagnosis, but you see traits of that diagnosis in relatives. And with an eye towards studying that phenomenon, in the early days, in the 90s and 2000s, lots of people developed so-called autistic trait scales. And those are continuous measures, not unlike height or IQ, that effectively estimate a number, that put a number on people's social behavior. Since it's an autistic trait scale, usually more autistic traits are associated with the higher end, and higher numbers are therefore worse. But it's continuously distributed, and you don't just see people with ASDs at one end and then a gap and a whole bunch of controls who are completely identical to each other. But you actually see a distribution of the population that's quite continuous, typically rather normative, where there's no clear distinction at which point, or to indicate where people should be considered categorically affected. So one of these measures is the Social and Communication Disorders Checklist, which has been used in various studies, but this one's the Avon Longitudinal Study of Parents and Children, which is a large cohort study of typically developing kids in the United Kingdom. And there was a GWAS done of it, of about 8,000 kids, or excuse me, the measure was using 8,000, but the GWAS was about 6,000 kids whose parents filled out this form, and the kids were assigned a number for traits of social communication disorders, where higher scores similarly indicate more impairment in social communication domains. So what we did is we used these polygenic genetic tools to estimate the genetic relationship between ASDs as they are diagnosed and social communication variation in everyone in the population, specifically 8-year-old kids in this case. And a genetic correlation, which is what we estimated, just gets at the extent to which two things are genetically related. So a genetic correlation of 0.3 would suggest that two things share about 30% of their genetic influences. And that's what we found here. We used results from two large common variant genetic studies, GWAS studies of autism, and looked at the association between the genetic influences on yes, no, do you have autism, and the genetic influences on what's your quantitative trait social communication phenotype in the general population, the influences on how people vary typically in the population in terms of their social communication ability. And you see about 30% sharing, which is just to suggest that there is significant and substantial genetic overlap between the influences on autism and the influences on social communication behavior in the population as a whole. And that degree of sharing is numerically quite similar to the amount of sharing between obesity and type 2 diabetes in genetic space. And we did something similar with de novo variants. We asked whether, if you look at de novo variant rates in controls, people who do not have autism, do you similarly see an association between their social and communication and daily living skills abilities and de novo rate in the population? And that is in fact what you see. This blue curve here is the association between de novo rate and functional ability and controls, where higher de novo rates are associated with lower functioning. And the red is the association between de novo rate and functioning, in cases. And in this region where both cases and controls can score, these would be the higher, and I'm using the term function here because this is actually, the Vineland is a measure of functional impairment. So cases with the highest functional abilities and controls with the lowest functional abilities overlap. In that region where they overlap, there isn't a distinguishable difference in their de novo rates. So neither phenotypically nor genetically is there often a clear distinction between cases and controls. And this is useful to consider in terms of genetic analysis because it changes the model through which we think variants are creating risk, and it also introduces a possibility of using control data, using phenotypic and genetic data from people in the general population to better understand the types of severe neuropsychiatric disorders we're interested in. And I think with that, we're moving to questions.
Well, thank you very much. This is Joanne Geer again, and I encourage those who have questions to enter them in the box. We have a couple of interesting questions here, and we have plenty of time to delve in. I find this absolutely, certainly fascinating, and Dr. Halliday and I did a program 5 or 6 years ago, and there wasn't nearly as much data available during that time. Dr. Halliday, could you speak a bit on how the industry, how the study, how the investigation of autism has changed over the last few years?
So, yeah, I mean, I can again reiterate what both Dr. Robinson and I have said earlier, which was, you know, I would say 10 years ago, the face of autism was those more severely affected, for lack of a better word, those with lower IQ, with more severe repetitive behaviors. They were the ones that actually, you know, were recognized as having autism. If you look at even the epidemiological data over the past 10 years, you're seeing more and more cases where the IQs are higher, the verbal abilities are better. They still have those core behavioral deficits in autism, but we're seeing a lot more differences across the spectrum, so to speak. So the slide I showed, which I stole it from someone, but I'm sure I could probably redo it, where, you know, you had the different domains, and you had some that had, you know, either nonverbal or, you know, low IQ, to those that had very high IQ and were you know, very verbal. So, you know, I think our understanding of that has really shaped how we're looking at genetics. I mean, in the very beginning, genetics was autism, yes or no. Did somebody have autism or did they not have autism? Now, as Dr. Robinson pointed out, we're kind of thinking about the overlap with other disorders, about subclinical features and what the genetics are of that. And I think that just recently in the past couple of years, that's led to, okay, now what do we do with that data? And that data means, you know, more targeted interventions. As research landscape has changed, there's been more interventions. There are advances in technology and advances in pharmacotherapy that have really helped many individuals with autism that just weren't there 10 years ago. So I think it's, we're moving kind of past genetic discovery and then on to how does this fit in with the bigger picture.
I think that's really interesting. That seems to be a pretty fast application of research. Would you like to speak some more about those changes in the interventions and what research is being done about their effect?
So it's really a kind of a mixed bag. So we know that behavioral interventions, and we've always known behavioral interventions are the most effective, but now researchers are finding that it's not just the behavior, that if you kind of pair behavior with either technology or with pharmacotherapy, that those interventions can get better. And that things like early behavioral intervention, which is amazing and actually does provide the best outcomes, doesn't work for everybody. And so, you know, we always encourage, or, you know, advocacy organizations always encourage people to get early intervention. For many, early intervention doesn't have the magic, you know, isn't the magic key that everybody had hoped it would be. So if we can identify things that would enhance those interventions, that would be great. There was actually a recent study that looked at the combination of augmentative technology, so iPads. So many people without any verbal ability or minimal verbal ability use iPads. And so iPads didn't exist 10 years ago, so here's a huge advance that has really helped people with autism. By adding iPads onto behavioral interventions that improve communication, not just speech, the outcomes were better than that with either alone. So it's not just about one or the other, it's about pairing them to be together. Now imagine how hard it is to design a study looking at one or two interventions at a time when people with autism are really in real world. I mean, you can't say to somebody, okay, well, you have to stop your ABA because we're just going to put you on this new drug. That's just never going to happen. So So the new kind of treatment studies really have to be by design combination studies where more than one intervention, a lot of times more than 3 interventions, are being studied at the same time.
Thank you. Dr. Robinson, would you like to add anything to that question about how the interventions are? You may not be on that side of the coin, but are you involved at all with the intervention piece?
I am not on that side of the coin. I think once data, again, it's in terms of integrating genetic data, it goes back to a substantial big data problem. So eventually there will likely be detectable differences in intervention efficacy based on genetic background, but it's going to take very, very large sample sizes to start to see that.
Are you seeing a larger or increasing willingness of families to engage in various kinds of studies to be able to collect more data?
I think Alicia might be the best, yeah.
I don't know if I can answer that. I would say that yes. I think that a lot of families get exhausted. I mean, so you look at this study, this project called the Interactive Autism Network, and this is this online registry that was started about 10 years ago, where people go in and they do everything online, right? So they enter in their own experiences. They enter in standardized instruments for their behavior, their child's behavior, if you're an adult, you can answer this. If you're a cognitively able adult, you can do this. Or if you're the parent of a cognitively able adult. And they ask just tons and tons of questions, and this has been just, as this data set has gotten bigger, it's had a huge impact, not just in research discovery, but in public health. I do think that and have heard from Ian, that there are a group of people that just fill this out and are done, and some that really will go back in and fill out every single questionnaire, and when a new one comes up, they'll fill it out. We are seeing, though, because of the need for big data, that there's kind of an interesting shift now. So whether or not you get a lot of data on fewer people, because you're going to get fewer people that are going to be willing to put all that time and effort into answering all sorts of different questions about all these different features of autism in themselves and their family, or less information on more people. So something that just popped up in the last week was the Simons Foundation recently launched this new initiative called Spark, and the idea is that 50,000 people in this online registry and get their DNA through saliva, but the amount of data that they're collecting is somewhat minimal. And the idea is that you can get more people to respond if you ask them less and less questions. And so I think that it's an interesting dichotomy that, you think more and more about, like, wouldn't it be great to know about this group of people who has this genetic mutation, and let's ask them, about their family history and their experiences and all of this, but at the same time, you don't get 50,000 doing that. You get 50,000 by being really good at asking just the right questions and the right number. So I think it's good, though. I mean, I think that that's okay to have that dichotomy, that there are different approaches. Certainly you can be a member of more than one research study and become heavily invested in one, and you don't have to be invested in others. The important part is that if you are a family that's affected with autism, that you do participate in research, and some of the ways are now online, which is a lot easier than dragging your child into a clinic for different assessments all the time. So.
Thank you. In some of the other research conversations we've had, the issue of the levels of consent for the use of the samples and the data has been interesting. And are you seeing a shift where, it was, yes, you may use my sample for research, but now the question in many cases is, are you giving permission to use the data from the sample, right, as it goes on and time goes on and more can be gleaned. Are you seeing any difference in those shifts, any shift there in the whole consent picture when people get engaged in these studies?
Well, Elise could probably talk about the genetic studies specifically and the consents needed, but we are seeing, some studies that their original consent was, okay, you can use it for research purposes. You can use the data set, you can use the sample for research purposes. And now as technology is getting better, it's now becoming, well, not just are we going to look at, these genetic assays that can't identify you. Now there's these genetic whole genome sequencing that can identify you on a person-to-person level. And so a lot of studies are having to go back and reconsent to make sure it's okay with the families that they, that they actually are okay with having not only that genetic data shared, because although it takes a lot of time and a lot of effort to take somebody's genetic their genome and then match them to a person, it is possible. So I think people have gotten a little bit more wary of that, but I also think that people are getting more and more wary of this idea of having their data in some sort of electronic form. And this is big data in general, right? And I'm sure there's been questions about ethics and consent in these big data webinars, but there have been some studies that have shown that there's greater concerns now because you put your data somewhere electronically and the next thing you know, someone's hacking into your data, into databases. I mean, they could be stealing your credit card number, next thing you know, they could, and this is a fear, I'm not saying this is a reality, this is a fear that they could be stealing personal information having to do with your health. And I think people are almost equally split as to whether or not they're comfortable having data shared electronically or just in paper, which I think poses a pretty high, it poses a challenge for the Affordable Care Act, where a lot of things are being moved over to electronic forms. And I think electronic forms are actually better, but I just want to recognize that, there are some concerns from the public about that.
Thank you. Dr. Robinson, do you have any thoughts? You want to address that side of the coin on consent and how it affects the work that you do?
Sure. So we deal with this a lot because the Broad is a very large sequencing and genotyping center, and we're a hub for a lot of various consortia and big data activities. So what you often see is we receive samples from a small study that is going to be a new part of a consortium effort. And so we have to effectively reuse data from a study that was perhaps, as it was initially consented, small, to make it part of something big. And usually that process is reasonably straightforward, and as long as the consent is written openly enough to involve and broad genetic analysis isn't a problem. Occasionally you have to go back and reconsent if you're going to use it for another purpose, but as genetics has really at this point nearly universally adopted big data protocols, the studies are now being planned with that in mind, regardless of their initial size.
Great. So we have a couple of questions here. Let's see. Do the big data models include information on so-called environmental or external factors?
I could speak to that a bit. Okay. So there are a couple reasons that environmental epidemiology hasn't as quickly adopted big data models that genetic epidemiology has. And the first is that simply the field doesn't have the same tradition of data sharing and consortium approaches. And those things are thankfully changing, because for the same reason it's going to take many, many people to identify the relationship between genetic risk factors and autism, it's going to take many, many, many people, if not even more people, to consistently and reproducibly associate environmental risk factors with autism. And that's because environmental risk factors are just harder to study, and it takes more people to sort out the various confounding things that could be going on. The second reason is there is legitimate additional complications on account of identifiability of people when it comes to deeply phenotyped data sets and environmental risk factors. People discuss identifiability from genetic data frequently. However, it's much easier to identify someone from a deeply phenotyped data set from the phenotype data. For example, the Avon longitudinal study of parents and children that I was referencing, the reason those data could never be publicly shared is because of the enormous contribution all of these families have made in terms of providing an incredible amount of data at multiple points every year for 20 years of their child's life. That phenotype data is very easily identifiable in aggregate. And so data like that, or data like the giant registries in Scandinavian countries, does have to be siloed to a greater extent than genetic data because it is more personal and more private. So various computational techniques are being developed to analyze those data in the cloud in-house so that they never actually have to be shared and can't get out into the world. But those technologies and kind of the field's desire to share and grow are kind of developing at the same time.
Well, terrific. Well, we have one more question, and then we'll ask you for some final comments. Let's see. Have researchers started to consider epigenetic contributions to autism in the sense that the epigenome is thought to play a role in pre- and postnatal development?
So this is Alicia. I can answer that. I hope I'm still on.
You are.
Oh, great. So yes, absolutely. There have been a number of new studies led by different researchers. I think some of the more prolific are at Johns Hopkins University that have looked at the epigenome in people with autism. As a nice kind of segue to that, Autism Science Foundation and Autism Speaks and the Escher Fund for Autism organize a webinar every few months about a different topic in epigenetics and autism. And on May 4th, we'll be doing one around imprinting. So imprinting is one of those phenomena where you get a copy of your genes from your mom and one from your dad, and sometimes one of those is silenced, and so you only get gene for different genes, not just completely across the board, but for different areas of the genome or different genes, you only get activity from one parent or the other, or you get two copies from one parent. So there are many disorders that come from imprinting, and so we're going to talk about how that affects autism. But I think it's an emerging field, and it certainly would explain a lot about how genes and the environment interact, but it's, again, an emerging area of study in autism.
Terrific, and we'd be happy to share the information about that webinar with our community. I'm sure there'd be a lot of people interested in that. I'm going to thank you and ask you for any final comments you'd like to make, and maybe you could talk about what you see coming down the pike, what's coming next in research around autism and using big data. Alicia, would you like to go first?
Oh, okay. I was just going to say, actually, the annual meeting for autism research is coming up in May, and so there'll probably be a lot of interesting discoveries about big data there. I think that some of these large analyses, these consortium-wide data, whether you're combining a lot of different data sets or you're collecting data from a large group of people anew, is really going to lead to some tangible answers. And so while some of them may not be found for a couple years, it's still exciting to think about how far further science is now than they were even 5 years ago.
Yeah, I completely agree, and I think moving forward, now that we have this abundance of genetic association to autism, people will be using those results to start understanding underlying biology, understanding what types of proteins and pathways these genetic risk factors are influencing, and people will be, as I discussed, using the genetic results to better understand phenotype, better understand heterogeneity, and better understand relationships between disorders. And my additional comment would be, please feel free to email me at any of my easily Googleable email addresses if there are any additional questions.
Thank you, Dr. Robinson, and thank you, Dr. Halliday. This has been really terrific. This session has been recorded, and within a couple of days it will be available through our website, and we'll be sending out a note for everyone. Thank you. Have a great day, and see you next time.